make sure that people can hear me. Hello, hello, hello. At least I hope so. Okay, good, good. Thank you. Because I've got a headset on, so I wanted to make sure I could be heard. Awesome. Thank you. Okay. Good morning. Almost afternoon here in Texas. So go ahead and get started here in just a second. Awesome. Thank y'all for letting me know that you can hear me. I appreciate that. All right. So we have a lot of ground to cover because Unit 2 has 11 topics in it, and a couple of them are repetitive. So there's probably not going to be as much content with those as there are for some. So like I said, I'm 2 minutes early, but I am going to go ahead and just get started. So this is the Unit 2 cell structure and function review. Um, you will notice there's going to be just a little bit of a lag between the stream and the slides. Um, and so, yeah, you're very welcome, Christopher. Thank you for being here today. But there is going to be a slight bit of a lag between the stream and the slides. And so, yay, internet. So I just want to say hi. I'm Lee Ferguson. I teach at Allen High School in Allen, Texas. And thank you all for being here today. I also want to say thanks to my moderators, Jamie Castle from Westchester, Pennsylvania, Clifford Nefrata from Phoenix, Arizona, and Danielle Tyeski from Dallas, Texas. And so let me kind of explain a little bit about some housekeeping items, just FYI. The slideshow is linked below, and it is set to view as a web page. If you actually want a copy of the slideshow, just reach out to me through my Twitter handle, and I'll be more than happy to provide that for you if you are a teacher. The schedule of future reviews is also linked below in the description. And students, if you have questions for a future review, then the question submission form is below, and it's helpful to the presenters if you submit your questions about a week in advance so that the presenters have a chance to see what questions you have so they can maybe build their presentations around your questions. And then students, how to use this review. It's reviewing the big concepts associated with cell structure and function. It's not meant to reteach the entire unit. This is a unit that is typically taught in the long school year over 3 to 4 weeks, and I have an hour. And so I'm really going to kind of speed through and hit some big ideas associated with each one of these concepts. I also want you to think about how questions over this content might be asked. And this is also to kind of supplement any review materials that you may already have. So let's look, sorry about that, hit the wrong key. So there's some concepts that we need to go over, and there are a lot of them. And so there's cell structure when we talk about the subcellular components, the organelles. You've got to be able to describe processes those organelles are involved in, individually and together. Cell structure and function, be able to make claims about structure and function. Cell size, you need to be able to perform calculations about cell size and represent relationships about cell size. Membranes, so be able to describe characteristics of membranes from their visual representation. So if they give you a model of a membrane, you've got to be able to describe that. Membrane permeability, where you propose an investigation based on an evaluation of some membrane permeability experiment. Facilitated diffusion, where you're going to predict changes that could occur in a system where facilitated diffusion is happening. Tonicity and osmoregulation, everybody's favorite, where you construct a graph, plot, or chart using data about tonicity or osmoregulation, and I'll talk more about that when I get to that section of the presentation. Mechanisms of transport. How are materials transported across cell membranes, whether it's active transport or passive transport? And finally, cell compartmentalization and origins of cell compartmentalization. With regard to cell compartmentalization, you want to be able to predict the cause or effect of a change or disruption to a cell because of the compartmentalization characteristic. And Finally, the origins of compartmentalization. Be able to support a claim with evidence demonstrating the occurrence or the presence of compartmentalization. So think endosymbiosis there and what evidence we have that that has actually happened. So let's hit the ground running. We've got the subject 2.1 is the cell structure. It's all the subcellular components. So essentially, you've got to know the different organelles. Now, you're going to notice that in the next list that I show you, nucleus and nucleolus aren't listed. That is because those get discussed in far more detail in cell division, mitotic cell division, and in nucleic acids. And so they're not so much focused on here in this topic. I'm not saying they're not important, but that is where the focus of those organelles tends to be, is in those other topics. You've got to be able to describe the structure or the function of each organelle. And so the organelles that you need to be really familiar with are going to be the ribosomes, rough ER, smooth ER, your Golgi body, your mitochondria, lysosomes, vacuoles, and chloroplasts. Now, I'm going to give you some hints about how to talk about these things, because we tend to want to talk about them using analogies that we learned maybe when we were in middle school. And so I want to kind of let you in on some things as a reader, because I'm also an AP reader. So I'm one of the, 750 people that sits in a very cold, very quiet room normally, and grades your AP exams. Now, this year, unfortunately, we don't get to do that. We'll be doing it at home, but we're still going to be marking your exams. So you need to be able to describe what characterizes each of those organelles. So, for example, a ribosome is made of rRNA and protein. Don't use analogies to describe the structure. Okay, describe the actual structure. Remember, it is a college level course. Therefore, the expectation is that you're giving a college level answer. So, for example, the Golgi body is not a stack of pancakes, because that doesn't tell me what it actually looks like. It doesn't tell me what its actual structure is. So what you do want to say, though, is that the Golgi body is a highly folded set of flattened membranes that are specialized for protein modification, packaging, and transport. So please get away from using the analogies that you've always, used in order to learn what these things are and how they're built, and tell us about the actual structure. And the same thing goes for function. And so you have to be able to describe what each organelle does and how they interact with each other and what those organelles are responsible for. And so in talking about the function, you've got to be able to describe what the function of each of those organelles in 2.1 is. And so, and I apologize for not being able to go over every single function. Remember, I have an hour to do this, and I have, a lot of other ground to cover that I think is far more important to review with you. And so, be able to describe the function of each organelle. So, for example, the double membrane of the mitochondrion helps to compartmentalize the reactions of aerobic cellular respiration. And don't use analogies. Again, the mitochondria is not the powerhouse of the cell, right? Because I know when I ask my students what a powerhouse is, they really can't tell me. And so, please avoid using those analogies that you've always known and that you've always learned and that you've always heard. It is responsible, though, for producing ATP in aerobic respiration and, the oxidation of the pyruvate that comes through. So, or actually, yeah, oxidation of pyruvate, but also oxidation of NADH. So please make sure that you're using actual descriptions rather than the analogies that we've learned to describe those things. And so hopefully that makes that very clear to you as in terms of like what you should be doing. Now, you do have to know some pretty specific knowledge about the chloroplast and the mitochondrion. And I know some teachers focus on this here, and I know some teachers focus on this in the energy exchange unit that they teach, where they talk specifically more about the mitochondria and the chloroplast and their roles in, exchanging energy with their environment. And so It does fall under this topic, so I'm going to talk just briefly about it here. And so, when you're learning about the chloroplast, remember that you have to know specifically about what the thylakoids are responsible for, how there's chlorophyll embedded on those thylakoid membranes, and this is where the light dependent reactions of photosynthesis occur. And then there's the stroma, which is the liquid component of the chloroplast, which is where the Calvin cycle occurs, or the light independent reactions of photosynthesis occur, and how, a grana is a stack of thylakoid membranes. And so you do have to know some pretty specific things about the structure of the chloroplast and the function of those particular structures. And the same thing is true for the mitochondrion. You need to know that those folds on the inside, the cristae, are part of the double membrane structure of the mitochondria that help to increase surface area for the reactions of the electron transport chain, and that the matrix is that liquid component where the concentration of hydrogen ions increases during cellular respiration. And so you do need to know, some pretty specific portions, of each of those different organelles. And then also, of course, for the mitochondria, the intermembrane space. And so please make sure that you review those specific components of the chloroplast and the mitochondrion, because you are going to be expected to know, the specific functions of each of those parts of those organelles. Okay, so awesome. That's right, endosymbiotic theory. So let's talk a little bit about cell size, right? So when we talk about cell size, this is, probably where your teachers may have done a lab with you where you learned about how important it is that cells be small. And the reason why cells have to be small is because the surface area to volume ratio is a pretty important relationship in terms of maintaining, homeostasis for a cell and the survival of that cell. And so if you look at the two diagrams that I have there, and I apologize for them being kind of small, if you look at the actual presentation on your computers, then it shows up a lot larger. Let's start with the diagram on the bottom there, where you've got the same sized cube, but cut several different ways. And notice that the smaller the cubes get cut into, the more surface area there is for the same amount of volume. And so that ratio of surface area to volume gets sufficiently large. And if you look at the graph above, because remember, cells aren't square. I was going to say, normally cells are not square in shape. In fact, most cells are not square in shape. But the graph above shows the relationship between surface area and volume for a sphere. And so notice what the relationship is between the surface area to volume ratio and the radius of that sphere. And so one of the things that you need to be able to do is to explain that relationship between those two variables. In other words, what happens to the surface area to volume ratio as the radius of the cell increases? What happens to it as it decreases? And read the graph carefully, because notice as the radius gets larger, that surface area to volume ratio gets smaller. And what this implies for the cell is that it makes it a lot more difficult for the cell to transport materials in and out, because the larger the cell is, the harder it is for the cell to either move in necessary nutrients so that it reaches all the parts of the cell, or to move out wastes. And so those wastes may accumulate and ultimately kill the cell. So it's really important that the surface area to volume ratio for a cell be sufficiently large. And so, the relationship you need to make sure that you understand, besides the surface area to volume ratio and radius if the cell is spherical, is that as the volume increases, the surface area decreases. And that cells have ways of, adapting and being adapted to help themselves achieve a higher surface area to volume ratio. And so one of the things that can happen is that cell membranes can fold. And so if you notice, with the previous slide with the mitochondrion, notice that was membranes on the inside were way folded up, right? Cells also can have extensions, and I didn't write that on this slide, but I want you to notice the picture that's there. It's a picture that shows, epithelium in the gut, right? And your gut is your intestine, specifically your small intestine, used for absorbing nutrients from the foods that you eat. And so, in order to absorb as much of the nutrients that get digested by your duodenum, you have all these little tiny microvilli on top of the villi on your small intestinal epithelial cells. And that increases surface area as well for absorption of nutrients. And so the other thing that cells can do is once they get to a certain size, in order to increase their surface area to volume ratio, they undergo cell division. They can divide themselves into two smaller cells so that they can increase that surface area to volume ratio. And so that helps also explain why, for example, the cells of an elephant are the same size as your cells, even though the elephant is hundreds of times larger than you are. Just because the animal is large or the organism is large does not mean that their cells are large as well. Their cells have to be small too, even though they are made of just a lot more cells. So be aware of the relationship between surface area and volume in cells and why that is so important for the survival of the cell. Now, with cell size, there's some math that you're going to have to do, potentially. And so there's a bunch of different formulas associated with cell size, and the formulas are all right here. Please don't memorize these at all, because you need to know how to apply them. It is more important for you to be able to apply these formulas than it is for you to memorize them. Remember, you do have access to a formula chart, and so spend your time knowing how to apply these formulas rather than memorizing them. And so we have volume of a sphere, volume of a cube, volume of a rectangular solid, and volume of a cylinder. And the reason why all of these formulas for volume are important is because cells can take these different shapes. They can be spherical, they can be cubic, they can definitely be rectangular solids. Think about plant cells. They can be cylindrical. Lots of animal cells are cylindrical in shape, like, for example, epithelial cells in the gut. But then you also have your surface areas. Surface area of the sphere, surface area of the cube, surface area of the cylinder. And again, don't memorize these formulas because you will be given a formula chart. And so it is important that you know how to apply these formulas rather than just being able to memorize them. All right, let's talk about membranes. And so membranes are critical in that they allow cells to keep a separate internal environment that is different from the external environment. Okay, remember that a cell membrane is made up of phospholipids, it's a bilayer, and that it's got proteins that are within it, it's got glycoproteins, it's got cholesterol and glycolipids embedded throughout. Now, please understand that, remember that your cell membrane is what they call the fluid mosaic model. And the reason why it's called a fluid mosaic model is because all of these components, these protein components, these glycolipids, these cholesterol molecules, they can move within the matrix of the membrane itself. And so the membrane is a very dynamic, structure. It is not static at all. It's very flexible. And when you realize what the membrane is responsible for, that flexibility is pretty important. And so understand that the fluid mosaic model of the membrane and its construction allows it to have the properties that it does for moving molecules in and out of cells. And so, that bilayer has two layers of phospholipids, and those phospholipids, as Ms. Castle described yesterday, are what we call amphipathic. In other words, part of the molecule is hydrophilic, part of the molecule is hydrophobic. And what does that actually mean? Well, the hydrophilic head is polar. Notice that it's got, a phosphate group right there. And that phosphate group is negatively charged. And so that hydrophilic head is pretty important because it's going to be what interacts with the aqueous environment of either the external environment of the cell or the internal environment of the cell. And then you also have the hydrophobic tails, which are fatty acids, those nonpolar fatty acids. Those fatty acid tails are going to face inward toward each other. And the reason why your phospholipids arrange themselves typically in, that head orientation, head being oriented out and tail being oriented in, this is the least, or it's got the lowest energy state, right? And so this is the most energetically favorable arrangement for these molecules. Like it's too that's the best way to say it. It is not energetically favorable for the heads to be pointing in toward each other. It is energetically favorable, though, for the tails to be pointing in toward each other. Because remember, the external and the internal membrane, or the external and the internal environments of the cell are both aqueous. They're both water-based environments. And so, the water interacting with the phospholipids is going to be attracted to the phosphate heads, partly because of hydrogen bonding. Because remember yesterday, Ms. Castle explained that hydrogen bonding is not truly bonding, it is just an intermolecular force between a hydrogen atom and something that's electronegatively charged, and phosphates are negatively charged, they're electronegative. And so the water is going to interact with that, but there's nothing on the fatty acids that the water is going to interact with because they're nonpolar, they're not charged. And so just remember that the reason why the membrane orients itself this way is because it's an energetically favorable arrangement, and so the phospholipid heads are going to interact with the aqueous environments that are either inside the cell or outside the cell. Now, the membranes also have proteins that are embedded within them, and those membrane proteins can be hydrophilic and have polar side chains. And typically, the membrane proteins that are interacting with the environment are going to be hydrophilic. And for the same reason that the hydrophilic heads are going to interact with the aqueous environment, if it's hydrophilic, it's going to be able to interact with that watery environment. But those membrane proteins also can be hydrophobic. And in fact, if they embed and anchor themselves into the membrane, they have to have a hydrophobic portion because the hydrophobic amino acids can seat themselves into the hydrophobic portion of the membrane. In other words, they're going to be able to associate themselves with the fatty acids. And so the portion of the membrane that's like embedded in the fatty acid portion of the membrane is going to be hydrophobic, because if it weren't, it wouldn't seat itself. Like there was no way that the protein would be able to maintain its location in the membrane if part of it were not hydrophobic. Now, there's a question about why can certain things pass through. That leads us to the next part of our presentation about membrane permeability and the fact that the membrane itself is semi-permeable, and the structure of the membrane allows for that semi-permeability or that selective permeability. Because remember how I said that the membrane is dynamic, it's not static, it's constantly moving. There are some small tiny pores within that structure that we don't really ever talk about, but they are there. I know I talk about it when we do the Piglo transformation lab, because one of the parts of that lab is that you have to heat shock the bacteria, and the reason you heat shock the bacteria is to kind of force the pores of the bacterial cell membrane apart so that the plasmid that you introduce to it can get through. And so know that there are small pores in the membrane as well, not just the channel proteins, not just the transport proteins, not just the glycolipids and the glycoproteins. There are just small pores because the lipids do have some small little tiny gaps in between. But other molecules can pass through the membrane, and how they pass through is going to be based on what characteristics those molecules have. So for example, if it's a small nonpolar molecule like oxygen gas, carbon dioxide gas, nitrogen gas, then those molecules, because they're tiny, they're going to be able to pass right through and go directly between the lipids themselves. But if they're large polar molecules, they need some help because they're too big or they have a charge. And so for example, glucose, sucrose, any of your sugars, glucose, sucrose, fructose, those sugars, they're kind of large. And so they need a little bit of extra help. So they're going to pass through a channel protein. But ions like potassium, sodium, calcium, chloride, all of those, they have to go through a channel protein because they have a charge. And so those molecules are all going to need a little bit of extra help in order to get through the membrane. Well, then you have polar uncharged molecules like water. And water can pass through the tiny pores in the membrane, but large amounts of water typically pass through aquaporins, which are specialized proteins that water passes through. Other polar uncharged molecules can pass between the lipids. And finally, large nonpolar molecules. I think that Zachary had asked a question about hormones. That could be considered a large nonpolar molecule. They can pass through, but they're going to come through really slowly through the lipids because they do tend to be pretty large. And so, for example, your lipid-based hormones, for example, because there are some hormones that are protein-based, but your lipid-based hormones would be something like this. For example, the sex hormones. They'll bind to a receptor on the outside of the membrane, and then because they are technically lipids, can pass through the lipid membrane, but they're going to pass through kind of slowly because they're giant and huge in comparison. And so I hope that addresses that question about why certain things can pass through, but it looks like my moderators caught it. So good job. All right, so the other thing that we've got to remember is that not every cell has just a membrane. You say we can't be exclusive. We have to remember that there are 3 kingdoms of life that have cell walls: plants, fungi, and prokaryotes. And so the cell walls not only are an important structural component, so say for example in plant cells, this is what provides the rigidity for the plant cell because the plant cell walls are made of cellulose. The cell wall is also a permeability barrier because there are certain molecules that can pass into the cell wall, but also certain things pass out. So for example, poison ivy leaves. The reason why there's oil on those leaves is because the plant cell can secrete those oils and then it can pass through the cell wall of the leaves, and then if you touch it, obviously you get a nasty rash. And so that right there should be proof alone that the cell wall is slightly permeable. And so that the cell wall is slightly permeable. So be aware that cell walls can be permeable. And one of the things I want you to also notice is that cell walls of fungi and bacteria and plants all have complex carbohydrates as a part of their structure. And the diagram that I've got here is slightly more detailed than you would ever really need to know, but know that each one of these cell walls is made up of complex carbohydrates, and they all perform a similar function. They all perform, the function of being semi-permeable and providing a barrier between the external environment and the internal environment of each one of those cells. And so, just make sure that you know that fungi cell walls are made of chitin, bacterial cell walls are made of peptidoglycan, which is a sugar protein complex, and the plant cell walls are made of cellulose. All right, so let's talk about transport, because membrane transport and tonicity seems to be the part of this unit that students struggle with the most. And so this is where I kind of want to spend more of my time, simply because it is the place where students seem to struggle. So when we talk about membrane transport, you've got 2 major classes. You have passive transport and active transport. And so let's spend a little bit of time talking about passive transport first. Remember that passive transport does not require ATP. You have materials moving with the concentration gradient. And so if you are ever asked to write about this, please don't ever just say the stuff is moving from high to low, because that doesn't mean anything to a reader. It should not mean anything to you. You need to say it moving from high concentration to low concentration, because then I'm clearly able to tell what you're talking about. But if you just say moving high to low, that really doesn't tell me anything. So be very clear when you're writing. Be very explicit in telling the reader what you want them to know that you're trying to communicate. Passive transport may use membrane proteins to move materials into or out of the cell. It really depends on what's being moved in or out. And so, when passive transport occurs, membrane proteins may be involved, but they're not always involved. And so remember, you've got two major groups of passive transport. You have diffusion, you have osmosis, and then under diffusion, you've also got facilitated diffusion, and we'll talk about that in just a second. But remember that diffusion is the passive movement of particles from high concentration to low concentration. So we could be talking about a gas, we could be talking about a solute, we could be talking about anything. But osmosis is a very specific type of diffusion because it is the diffusion of water from high concentration to low concentration across a semi-permeable membrane. And that's the key, is that osmosis involves water only. It involves a semi-permeable membrane, where diffusion does not necessarily involve a membrane, right? Because, like, for example, if I break a bottle of perfume in one part of my house, eventually that scent is going to spread throughout the entire house. And so that means that the gas or the aerosolized particles are going to spread through my home, right? Through from a high concentration area to a low concentration area. And so that's diffusion. But osmosis is going to be, let's say that I, cut lettuce for a salad and I put it in a salad bowl, and then I put, really salty olives on top of it, and then I put it in the fridge, and a few hours later I come back to eat my salad and my lettuce is all wilted and sad and my salad has a bunch of water in it and looks really unappetizing. That's osmosis, because the water has moved out of the cells that make up the lettuce across the membranes of those lettuce cells from an area of high concentration inside the lettuce cells to an area of low concentration, which is outside the lettuce cells. And it's all because of the olives that I put on the salad. Yeah, that actually happened to me once. It was very sad. But that is a practical application of osmosis. So lesson learned, kids, don't put olives on a salad and then put it in the fridge. All right, so membrane transport, with regard to active transport, it does require ATP. You have materials moving against the concentration gradient across the membrane. And so, in other words, now we're moving from low concentration to high concentration. This is always going to use some kind of membrane protein to move materials into or out of the cell. And this includes bulk transport like endocytosis and exocytosis. So let's talk a little bit more about each one of those things. So endocytosis and exocytosis, you're using vesicles to transport materials into or out of the membrane, or out of the cell, I'm sorry. And those vesicles are going to fuse with the plasma membrane. And this is typically used to transport large molecules either into or out of the cell. Like a really great example of endocytosis is white blood cells consuming, cells that have been infected with viruses, or bacterial cells, where essentially like the phagocytes engulf the offending cell and enclose it in a vesicle and bring it inside their cell, and then later will fuse it with a lysosome to digest it. And so that's a great example of endocytosis, which is, that particular type is called phagocytosis. Penocytosis is where you have, the cell is taking in small droplets of liquid into the cell with a vesicle. And then you also have what's known as receptor-mediated endocytosis, where there is a receptor on the outside of the membrane that is specific for a certain molecule that's to be brought inside. So a great example of this is cholesterol. And cholesterol will bind to a cholesterol receptor. Once the receptors on the membrane have achieved the number of molecules that they need in order for the vesicle to close, it will close. So the concentration of that cholesterol has to get to a certain level. Well, then once those receptors have bound all the cholesterol that they can bind, that vesicle will enclose itself and be brought into the cell itself. And those receptors will have the cholesterol binding to them inside that vesicle. And so that's a good example of, another way that molecules can be brought into the cell. And then, of course, exocytosis, where cells are excreting something or getting rid of a waste product. Like a really good example of this would be when a cell produces, say, like your digestive tract, your small intestine is producing not your small intestine, I'm sorry, your pancreas is producing the enzymes that are needed for digestion of proteins. They're going to package up those enzymes into a vesicle and excrete them into the small intestine through exocytosis. And so both of those are going to use ATP to help move those molecules into or out of the cell. Well, then you also have facilitated diffusion. And facilitated diffusion, I mean, if you think about the word facilitate, it means to help, right? And so facilitated diffusion is going to use membrane proteins to move bigger molecules and polar molecules into the cell with the concentration gradient. So it's still passive, right? It's still passive because we're not using ATP, but these molecules are so large that they can't just cross the cell membrane on their own, right? They have to have a little bit of extra help. It's kind of like the trucks on the road that have the wide load sticker on the back. They need a little bit of extra room. And so glucose is a good example of a molecule that is transported by facilitated diffusion. You also have sodium that can be transported by facilitated diffusion. And so know that these are going to be transported through the protein channels or carrier proteins. But the fact remains, it's still passive. We're still moving with the concentration gradient. And so you have no use of ATP here. And so one of the things that you need to be familiar with is that the membrane potential of a cell, which is basically it's a charge on a membrane. So for example, your neurons. Neurons have a membrane potential. And that membrane potential is maintained by the sodium-potassium pump, right? Like you've probably been taught about the sodium-potassium pump, how for every 3 sodiums that come in, 2 potassiums come out. And so the that charge on the membrane that allows you to have an action potential so that your neurons can fire, that's maintained by that sodium-potassium pump. And so part of those ions are going to flow in with the concentration gradient. And then some of those ions are going to flow out, or be brought back out through active transport. And so, that sodium-potassium pump, pretty important in terms of maintaining that membrane potential. And so notice the diagram, it shows you how the concentration of each of the different ions varies on each side of the membrane. And so notice that the sodium is high on the outside and potassium is low on the outside, but that it's reversed for the inside of the membrane. And notice that the ATP has to be hydrolyzed in order for that potassium pump to do its job. Now, tonicity. This is where students really tend to struggle. At least I find that my students tend to struggle without a lot of practice. And so tonicity and osmoregulation. You need to know that this is going to involve osmosis exclusively, right? And so water will always move by osmosis from areas of high concentration to areas of low concentration. And I want you to notice how I've highlighted the 3 different sentences here because they are all different ways to say the same thing. And so water, you can say that water moves by osmosis from an area of high concentration to an area of low concentration. But you can also say that it moves from an area of high water potential to an area of low water potential. And then you can also say that it moves from an area of low osmolarity, meaning that there's low solute concentration, therefore high water concentration, to an area of high osmolarity, which means an area of high solute concentration, therefore low water concentration. So all 3 of these are different ways to say the same thing. Be familiar with all 3 ways to say the same thing. Don't let any of these trip you up, and don't think that they all mean different things, because they don't. And so I wanted to make sure that I presented all 3 of these different ways to you so that you would know, oh, hey, they're asking me about this, or they're asking me to talk about this. And so these are also, like I said, 3 different ways that you could express the same idea. And so I think the more different ways you have to say the same thing, the better off you are. And so let's talk about what that means in terms of tonicity. So when we talk about hypertonic solutions, right? A hypertonic solution is a solution that has a higher solute concentration or a high osmolarity relative to the solute concentration of the cell. So if I take a cell and I put it into a hypertonic solution, water's going to move out of the cell. Now, having said that, I also need you to understand that when we talk about tonicity, osmotic solutions, that the tonicity is relative, right? Because you can have a hypertonic cell. So for example, plant roots. Plant roots, by their nature, are hypertonic. In other words, they have a high solute concentration, and the solute concentration in a plant root is going to be the stored carbohydrates, you know. And the reason why this matters for plants is because this is how plants get water. If the cells of the plant root were not hypertonic, water would not move from the soil into the plant cell. And so it's not just the solution outside the cell that can be hypertonic, the solution inside the cell can be hypertonic as well. And so think about it like this. Plant roots are hypertonic. This is why water moves from the soil into a plant root. Okay? So when we're talking about osmotic solutions, remember the concentrations of solute are relative to whatever's on either side of the membrane. And so there's your example of hypertonic, how a hypertonic solution would act in the cell. And I've explained about plant roots. And I may add that to my slide just to have another example for you. Hypotonic solutions have a lower solute concentration relative to the solute concentration of the cell. So in other words, the solution has a low osmolarity. In other words, low solute, high water concentration. If I put my cell into a hypotonic solution, water is going to move into the cell. And so a good example of an organism that has a hypotonic solution, or that lives in a hypotonic solution, would be a paramecium, this single-celled organism. Now, the paramecium is this freshwater, protist, so it's a unicellular little guy that lives in ponds and lakes and stuff like that. And what's really cool is that it's got a high water concentration inside its little cell, but the water concentration outside of its cell is higher than what's inside of its cell. And so in order to prevent water from moving in at too great a volume and to maintain equilibrium, in other words, to maintain the same concentration of water on either side of its membrane, it has this little contractile vacuole organelle that pumps out the water that comes in. And so when that water from the lake or the pond moves into the little paramecium cell, it immediately pumps that water out, because if it didn't, it would rupture, it would lyse. And so that little pump is constantly moving. So it's using a ton of ATP to move this excess water out of its cell. And I'll add that to this slide as well. Because I was going to talk about how organisms have adaptations for regulating osmotic solutions in their cells and in their bodies, but I'll go ahead and just talk about it as we go along. And so, if you live in a hypotonic solution, like if you're a single-celled organism like the paramecium, then you're going to have some sort of organelle or some sort of structure to help you maintain equilibrium. In other words, you're going to have some kind of way to maintain homeostasis. And then finally, you have an isotonic solution where the solute concentration on both sides of the membrane is going to be equal. It's going to be the same. And so one of the things that students often say is that, well, the water doesn't move. That's not true. Water is moving. There's just no net movement. So the water is moving back and forth at equal rates. And so net movement of water is zero. And so water is still going to move back and forth because, y'all, molecular motion does not stop unless it is absolute zero. And we can't achieve that temperature on the Earth. And so water is still going to move back and forth at equal rates. There's just not going to be a net flow. In other words, there's not going to be, more on one side than the other. And the system will have achieved equilibrium at this point. And so, I want you to understand that in an isotonic solution, the cells are moving water back and forth. The water's moving back and forth across the cell membrane. It's just moving at an equal rate. And so, for example, when someone gets an IV, and in a hospital, the concentration of solutes in that IV bag is roughly equal to the concentration of solutes in the plasma. So that, number one, the person doesn't become dehydrated, and number two, they don't become like over dehydrated. Excuse me. And so, just be aware of the fact that, your different osmotic solutions will cause cells to behave differently based on the solute concentration that's on either side of the membrane. And remember that concentration is relative, because it's not just the solution outside the cell that can have one of these characteristics. The solutions inside the cell can too. So, one of the questions that I was asked in the question form was, can we work some water potential problems? I'm going to go ahead and work one with you, but I'm going to also say that in the quantitative skills presentation on April 26th, there's going to be more water potential problems worked. And so I just wanted to make sure that you understood like which formulas you would need to know. And again, don't memorize these. You have a formula chart. You will be given access to a formula chart. But remember that water potential, for our purposes, is equal to solute potential plus pressure potential. So those little funny pitchforks, those are the Greek letters psi. So psi equals psi sub s plus psi sub p, where solute potential is psi sub s and psi sub p is pressure potential. And the solute potential of a solution is psi sub s equals negative ICRT, or as I always say in my class, Ickert, where negative I is the ionization constant, and how the ionization constant is determined is by how many ions the solute breaks apart into. If there is a sugar as your solute, then the ionization constant is always 1. Okay, it's always 1. And otherwise, it's going to be the number of ions that solute breaks apart into. And so, for example, if it were sodium chloride, it would be 2, right? But anytime it's a sugar or something that ends in ose, it's going to be 1, because those things don't ionize, okay? And so your concentration is the molar concentration of solution. And R is a constant, which is 0.0831 liter bars per mole K. Okay, and then temperature is the temperature in Kelvin. And always remember, for temperature in Kelvin, you have to add 273 plus the degrees C. One of the things that students always forget is to add the Kelvin degrees. So please make sure that when you're calculating temperature for the solute potential equation, that you add the 273, because that is something that students tend to forget. So if we look at the question here, where it says the student places a potato core into an open beaker, has a 0.55 sucrose solution, the temperature of the solution is 25 degrees C. What is the water potential of the sucrose solution? So we have to think first, which formula is the one that we need, and we're going to assume that psi p is equal to 0, and we have to think about why. So why are we assuming that psi p is 0? Okay, think about why psi p is equal to 0 based on what information you've been given here. Right, so remember, that's right. So because it's an open beaker, okay, good, Jenny. Because it's an open beaker, we assume that the pressure potential is equal to 0 because we're at atmospheric pressure, okay? It's not pure water. We've got a sucrose solution, right? If we have a sucrose solution, it can't be pure water. And so we have an open beaker, and it is at 25 degrees C. So we're really solving for psi equals psi sub s. And so we know that psi sub s is equal to minus ICRT. Okay, and so now we have to figure out, now what? Okay, so you've been given -1 because sucrose is the solution. Okay, you've been given the concentration of the solution, which is 0.55 molar. You've also been given the constant, which is always going to be the same because it's a constant, so that 0.0831 liter bars per mole K. And then you've been given the temperature, which is 25 degrees C. And again, remember, add 273 to that. And so 273 plus 25 is going to be 298. Okay. And to answer Alex's question, if it's not an open beaker, will you be given pressure? Yes, they would tell you that. Yeah, assume that unless they give you the pressure potential, that the pressure potential is going to be equal to 0. Okay, but yeah, they would tell you what the pressure potential was if it wasn't an open container. Okay, so there's all your givens. So all you got to do is plug it in. So if you set it up, you plug it in, you -1 times 0.55 times 0.0831 times 298, and through cross multiplication, you cross out every unit except for bars. Your answer is going to be -13.6 bars. Okay, and so hopefully that question wasn't too bad. Sig figs, they will tell you like what place to round to typically. Like they'll tell you round to the 10th place or round to the 100th place. So make sure that, if they tell you specifically what to round to, that you round to that place. Okay. And so here I rounded to the 10th place, even though my sample question didn't say so. But I went ahead and rounded to the 10th place. But yeah, typically you'll get told like what to round to. And so, what I'll also do later today is I will link a set of practice problems so that you can have those to practice with, because I know that some people really like to be able to practice this stuff, because I know that was something that was asked in the question submissions that we got. And so I want to make sure that you get the practice that you need. All right. So let's talk about mechanisms of transport super briefly. Okay, because this was a little bit repetitive. And so you have to be able to describe the mechanisms of transport. In other words, the forms of active transport, what can be moved into or out of cells using this method, how does this happen, what kinds of passive transport are there, what can be moved into or out of cells using this method, how does this happen, because when I looked at the course exam description, the CED, it was not real descriptive about what should be discussed here. And so I felt that those things had already been discussed in some detail earlier. And so I just wanted to kind of revisit and say, be able to describe these things. You've got to be able to describe these things. And so when you're asked to describe a concept or to describe, a process, this is what they're asking you to do here. So what can you move? How can you do it? And so when you're describing something, it's a what and a how is what you're looking for. And so make sure that you're able to do both of those things. And then compartmentalization and the origins of compartmentalization are going to be the last two things that we're going to talk about. And so compartmentalization kind of represents like the ultimate in membranes and transport, because compartmentalizing a cell allows it to do a lot of really cool stuff. Like it allows it to localize metabolic processes. So it allows it to have like special compartments for certain chemical reactions. It allows it to, increase its surface area. It allows it to specialize, right? Like this is how we get specialized cells, right? Because the first cells were bacterial cells that weren't terribly specialized, that weren't, very complex. And so the fact that eukaryotes came along about 2.7 billion years ago is what allowed the cells to specialize and to become more complex. And so compartmentalization allows cells to specialized, like I said, you've got, say for example, in a eukaryotic cell, you've got a nucleus that houses the genetic material. You've got the mitochondria that houses metabolic reactions. You've got vacuoles that store wastes or fluid or nutrients. You've got plastids, like in chloroplasts, that store starch, that have pigments for photosynthesis. You've got the Golgi body. You've got the ER, both the rough and the smooth ER. And so each one of those different compartments is super important in helping to, separate all these different chemical reactions from one another so that they're not competing with each other, right? And so, the compartmentalization and specialization of cells is pretty important. And then finally, the origins of cell compartmentalization, this is all about endosymbiosis. And so specialization, let me look at your question, Zach. I would say specialization, kind of both actually, in terms of, specializing into different types of tissues, but different organelles. I mean, if you think about it, the fact that cells in eukaryotes can compartmentalize is why humans specifically have 240 plus different types of cells, right? Because every different cell type is going to have, different different compartments in different cell types are going to be specialized based on what that cell's job actually is in the organism, right? And so, for example, your immune cells probably going to have a lot of lysosomes. Your mitochondria, or not mitochondria, but your muscle cells going to have a ton of mitochondria. Same with neurons, tons of mitochondria, lots of Golgi bodies. And so, even within cell, different tissue types, there's going to be specialization there as well. So I hope that kind of answers that question. But I wouldn't expect questions to be asked about specific tissue types without any kind of context. Because that's the thing, I'm seeing a lot of questions about immune systems and nervous systems and whatever, and you'll get background information given to you about those things. So, technically you could be asked a question about endocrine cells, but they would give you the background knowledge that you need to know in order to be able to answer that question. But really the overarching concept could be compartmentalization, right? Technically they could ask you about digestive system, even though that shows up nowhere in the course and exam description. But as long as they gave you the background information that you needed to know, and they, all of that, then they could ask a question about, increased surface area of the intestinal tract cells. And let's see, are mitochondria organelles? I guess technically, yeah, because they are subcellular structures. So yeah, they're organelles. And so if we talk about like the origins of compartmentalization, we know that endosymbiosis is the commonly accepted theory for how cells became eukaryotes, where a primitive prokaryote engulfed another prokaryote and brought in, say for example, for the mitochondria, aerobic prokaryotes that became engulfed by the other prokaryote, and then over time cyanobacteria that got engulfed that became the chloroplasts. Now, one of the things that I could see being asked, and when I say that, please don't think that I'm prognosticating or anything. Like, I don't know what's going to be on the exam other than biology, but I could see a question being asked about evidence that endosymbiosis has occurred, right? Like that's a really common thing that teachers like to ask, like, how do we know that this happened? And so, we have to be able to talk about the evidence that supports the claim that mitochondria and chloroplasts were free living. And so we have a lot of that evidence. We know that they both have double membranes. We know that they undergo binary fission, that they can only come from other mitochondria and chloroplasts. We know that they have their own DNA. We also know that they have their own ribosomes that are smaller than eukaryote ribosomes, right? Like prokaryote ribosomes tend to be 70S in size. Well, same is chloroplast mitochondria. And, eukaryote ribosomes tend to be 80S in size. So there is quite a bit of evidence that exists that demonstrates that these were free living cells before they became incorporated as a symbiont into prokaryotic cells. So we've got about 5 minutes left. And so one last thing before we end here. Know that prokaryotes don't have membrane-bound organelles, but they do have internal regions with specialized functions. And a great example of this is the nucleoid region, which is where the DNA kind of hangs out. Because notice, there aren't any organelles in my prokaryote cell. They're pretty basic. They have a membrane, they've got genetic material, they've got ribosomes, they've got a cell wall, they've got the cell, and the cell membrane, I think I said that. But the nucleoid region is kind of where the DNA hangs out. And so, prokaryotes are pretty simple. They also have, some of them have flagella, some of them also have cilia, but that's not shown here in this picture. So, I know that was an awful lot of material in a really short amount of time, but I wanted to thank you for being here today. I want to thank my moderators once more for fielding questions and cleaning up the chat for me. And if you have questions, find me on Twitter. My handle is @the And I wanted to wish you luck. I was going to say we've got a few weeks away yet, about 6 weeks away, and you can do this. I was going to say you can definitely do this. I was going to say I hope that you are staying safe and staying healthy. And that, take time every day to do something that is fun for you. Don't just school all day long, because this is not a normal way to do school. And so, right now life is anything but normal. So I hope that you're taking some time to appreciate something small every day. So have a great day. And like I said, stay safe, stay healthy. And you all are so very welcome.